Welcome everyone to your second lecture held online for Anatomy and Physiology 2, covering chapters 32 and 33 on the immune system. So just to introduce the immune system, it protects against assaults on the body, whether they're external assaults or internal assaults. And the real key is cellular identification, being able to identify whether or not a cell is good or bad. We will talk today a lot about antigens. Cell viruses and other particles have unique molecules and groups of molecules on their surface that can be used to identify them. These molecular markers visible to the immune system are called antigens. Our own cells have unique markers as well that we also call antigens that are embedded in our plasma membrane that each of our cells have, and we call this a self-antigen that is belonging to us as an individual, so we do not recognize it as foreign and we realize that we cannot attack it. Okay, so we have self-tolerance. Foreign cells or particles, however, have non-self antigens, and these antigens serve as recognition markers for our immune system that say you need to attack. This is non-self. There are two different types of immunity that we'll discuss today. One is innate immunity and the other is adaptive immunity. So innate immunity and adaptive immunity. Innate immunity mechanisms are sometimes called non-specific, and they're built in and ready for action. It's natural. It's what we're born with or what we develop naturally. It really provides us with our initial defense against infection and other assaults on the body. Then we have adaptive immunity. This is more specific immunity. These immune mechanisms develop later on over time as lymphocytes, which we'll talk about later, are activated to work against very specific antigens or abnormal cells and particles. We'll speak about these types of immunity in more detail throughout this lecture. So let's talk about lines of defense. So immune function, that is defense of the internal environment against foreign cells, proteins, and viruses, includes 3 layers of protection. Okay, we have our first line of defense, our second line of defense, and our third line of defense. The first line of defense is a set of barriers between the internal and the external environment. The second involves innate inflammatory responses, which include phagocytosis. And the third includes the adaptive immune response and the innate defense offered by natural killer cells, and we will elaborate on this later. Of course, tumor cells that arise within the body are not affected by the first 2 lines of defense and must be attacked instead by the third line of defense, which includes those natural killer cells. Okay, so first, second, and third line of defense, and we will begin elaborating on those now. Let's begin with innate immunity. So what is species resistance? Well, species resistance refers to a phenomenon in which the genetic characteristics common to a particular organism or species provide defense against certain pathogens. Pathogens are disease-causing agents. We are resistant as human species to certain funguses, for instance, that infect trees. Okay, so each species has their own set of resistance. The internal environment of the human body is protected by a continuous, what we call mechanical barrier. And this mechanical barrier is formed by cutaneous membranes, so the skin, as well as mucous membranes. Oftentimes referred to as the first line of defense, these membranes provide several layers of densely packed cells and other materials that sort of form a wall that protects the internal environment from invasion by foreign cells. Besides forming a protective wall, the skin and mucous membranes operate various additional immune mechanisms that we call chemical barriers. Substances such as sebum, which contains pathogen-inhibiting agents, mucus, in which pathogens can become stuck and then are swept away, and enzymes, which may hydrolyze pathogens. We also have hydrochloric acid that's found within our gastric mucosa, so within our stomach in particular, which may destroy pathogens as well. These all may be present to act as an innate defense for our human body's survival. Okay, let's move on to talk about the inflammatory response. If bacteria or other invaders break through our chemical and physical barriers, which I just spoke to you about, and that are formed by membranes and their secretions, then our body has a second line of defense, and this is called the inflammatory response. Tissue damage is going to elicit a host of responses that counteract the injury and promote a return to normal. So, for example, bacteria can cause tissue damage that in turn triggers the release of various inflammatory mediators from cells such as mast cells, which are found in connective tissue. Inflammatory mediators released from mast cells include histamine. Other cells can release cytokinins as well as prostaglandins. These mediators work to dilate blood vessels, but they also work to increase vascular permeability so that we can get more blood to the area. What is the purpose exactly? Well, the purpose is to help phagocytic white blood cells reach the affected tissue. Some of the characteristic signs of inflammation include heat, redness, swelling, and pain. Many of these mediators of inflammation that I've listed above are called chemotactic factors. That is, these are chemicals that attract white blood cells to the area through a process called chemotaxis or chemotaxis. So let's talk about chemotaxis and diapedesis. In this example, we have a neutrophil in the blood that is attracted by chemotactic agents or chemotactic factors that are released by a mast cell in damaged tissue or infected tissue. After adhering to the blood capillary, we call this pavementing. So it adheres to the blood capillary, and we call this pavementing. The neutrophil exits the capillary by the process of diapedesis, which we have already spoken about, but I'll remind you of what it is in a minute. And through the process of chemotaxis, which is movement directed by chemical attraction, the neutrophil migrates to the highest concentration of chemotactic factors. So you can see the neutrophil here adhering to the blood vessel. Through the process of diapedesis, it leaves and it's attracted to the area of highest chemotactic factor, which is really the site of injury. And this is where it can begin its immune functions. As you should recall from the in-class lecture, neutrophils have phagocytic functions. You should also recall that diapedesis is the process by which certain cells can pass directly through an uninjured blood vessel wall just to enter tissue. Okay, we'll talk a bit about phagocytosis now. A major component of the body's second line of defense is the mechanism of phagocytosis, which is essentially the ingestion and destruction of microorganisms or other small particles. There are many types of phagocytes and what I mean by talking about phagocytes is that it means that if a cell is a phagocyte, it is capable of the process of phagocytosis. In this image, we summarize the function of a phagocyte. When a phagocyte approaches a microorganism, they're going to extend what are called foot-like projections. They're actually called pseudopods toward it. Soon, the pseudopods are going to encircle the organism and form a complete sac called a phagosome. The phagosome then moves into the interior of the cell where a lysosome is going to fuse with it. The contents of the lysosome, specifically the digestive enzymes, are going to drain into the phagosome and destroy the microorganisms within it. So in looking at the diagram here, you can see that in the first step, a bacteria is encountered by a macrophagy. Within the macrophagy, it has its cell's nucleus, and it also has lysosomes, which contain digestive enzymes. In the second stage, we're going to see that the antigen is going to bind to receptors, non-specific receptors, because we're talking about innate immunity or natural immunity, which is non-specific. Then we're going to have those pseudopod formation, and it's basically engulfing through the process of endocytosis the bacteria. And then once the bacteria has been engulfed and enveloped, we're going to have lysosomes that are going to attach, these vesicles are going to attach to this enveloped off area so that I can release its enzymes and destroy the bacteria. And then finally, we're going to have the contents released, these non-harmful products. Now, many cells within our body are capable of phagocytosis, but the most numerous is the neutrophil, which is usually the first to arrive at the site of injury. It's going to migrate out of the blood vessel or out of the bloodstream through the process of diapedesis, and it's also going to form pus. That's an important feature. So neutrophils are going to usually arrive first at the site of injury, migrate out of the blood vessel through the process of diapedesis, and form pus. Because phagocytosis defends us against various kinds of agents, we classify it as innate defense. But, as you'll learn later, it also crosses over into adaptive immunity as well. Now, thinking back to the last slide, after the phagocyte digests the offending particle, the phagocyte will often process the protein and display bits of the protein, which we call peptides, on the surface of the phagocyte. Okay, so it's going to digest it, and then it's going to put some of those proteins on its surface, almost like a surface identifier. And these peptides can then be recognized by cells of the adaptive immune system, so referring to the other type of immune system. So now it's very specific. Now we just digested something that's foreign, and we're displaying what we've digested on our surface, and now we have a specific receptor should that foreign pathogen come into the body again, and these are called antigens. And cells that perform this function that I just spoke about are called antigen presenting cells, or APCs. Another type of phagocyte is called the macrophagy, which really just means large eater. A macrophagy is really a large monocyte, like we spoke about in class last week, that have grown to be several times their original size after they've migrated out of the bloodstream. And macrophages are an important antigen presenting cell, so doing that function that I had just discussed with you. Yet another type of important cell or phagocytic cell is called the dendritic cell, which is found in many tissues of the body that have contact with specifically with the external environment, such as the skin and mucous membranes. It's called dendritic because it has, as you can see in the diagram here, it has many different branches. The prefix dendra means branches. Okay, so again, these are found mainly in tissues that have contact with the external environment, such as the skin and mucous membranes. We've spoken a bit about local inflammation to a specific area, but systemic inflammation, so inflammation throughout our system, our body system, may occur when inflammation mediators trigger responses on a body-wide basis. A body-wide inflammatory response may be manifested by a fever, and a fever is just a state of abnormally high body temperature. So as an example, bacterial or viral infections that spread widely throughout our body can produce what's called a systemic inflammatory response syndrome, or SIRS for short. And it is specifically pyrogen molecules that trigger this fever response. Pyrogen molecules trigger this fever response when pyrogen is released from damaged tissues, or if it's introduced into the body, say in the form of a bacteria. Whenever you hear the word endogenous, we're just referring to originating within the body, and exogenous referring to originating from outside of the body. And pyrogen molecules trigger the fever response by promoting prostaglandin production, or PG production. PGs sort of reset the body's thermostat in the hypothalamus, and what they're doing is trying to set it to a much higher temperature, really trying to almost burn off the pathogen, so to speak. Aspirin, which I have listed here, as well as other cyclooxygenase inhibitors, which we call COX inhibitors, can reduce the production of this prostaglandin, which in turn will bring the body temperature back to normal. This is why many medications taken when you have a fever can make you feel a lot better, because a fever comes with symptoms such as aches, pains, fatigue, etc. And if you can reduce the fever-causing agents, then a lot of those symptoms from the fever go down as well. It is thought, though, that elevated body temperature is designed to facilitate an immune response as well as inhibit those pathogens, sort of killing them off. But the role of a fever is still widely debated amongst immunologists. Now, besides phagocytes, the body has an important set of cells that provide innate defense in the body, and we call these natural killer cells. Natural killer cells are a group of lymphocytes that kill many types of tumor cells and cells that are infected by different types of viruses. Natural killer cells recognize abnormal cells by using two different recognition receptors, which are very important for you to know. So we have one, a killer-activating receptor, which you can see here, and a killer-inhibiting receptor, which you can see here. Okay, it's these receptors that allow natural killer cells to recognize whether or not something's abnormal. The killer-activating receptor, so the killing-activating receptor right here, is going to bind to any of several common surface molecules that are found in cells. So you can see here that it can bind to a normal cell. So therefore, the natural killer cells can bind to any cell of the body as well as abnormal cells or foreign cells. But what's key is that if the killer activating receptor attaches to a normal cell, what's very important is that if the killer inhibiting receptor happens to bind to an MHC class 1 molecule, which stands for major histocompatibility complex, which is a protein, I should have said, then the killing action is stopped. So we might have this binding occur here, but in order to prevent actual attack, as long as we have binding here, then the body recognizes that this is normal. So this you could see is very non-specific, right? This natural killer cell is non-specific. It's binding to anything in the body, but provided that we have binding to an MHC class 1 molecule found on the normal cell, then attack will not occur. Or you can think of the killing action as being stopped. MHCs are surface proteins found on all normal cells, and they're very unique to each individual. Therefore, only abnormal and foreign cells fail to bind to this killer inhibiting receptor and are therefore killed by the natural killer cell. So you can see here the natural killer cell binds to an abnormal cell, but because there's no binding made by the lack of MHC class 1 molecule, then the killing process will begin by the natural killer cell. And the natural killer cell uses several different methods for killing cells, most of which progress to the formation or to the process of lysis. And lysis is breaking apart of the abnormal cell. And you should know that it is important that there is direct contact between the natural killer cell and the abnormal cell in order to go through the process of lysis. Lysed. Okay, so we're going to continue on now by talking about interferons and complement proteins. Several types of cells, if they are invaded by a virus, are going to respond rapidly by synthesizing the protein called interferon and releasing some of it into the circulation. The interferon that is released to nearby cells is going to signal these cells to enter what's called a protective antiviral state. In other words, the interferon is almost like sending out warning signals to other cells to help prepare for this incoming viral attack. And as the name suggests, the name interferon suggests, it interferes with the ability of virus to cause disease. Complement protein, this is the name given to each of a group of inactive enzymes that are later activated in a cascade of chemical reactions which results in a variety of different immune responses. Ultimately, the complement cascade causes what's called lysis or rupturing of the foreign cell, and we'll talk about this a little bit later. Complement proteins essentially complement or aid in the immune response. You can consider both interferons and complement proteins as a whole as protective proteins. So we have just finished talking mainly about innate immunity, and now we'll move on to adaptive immunity. Adaptive immunity is considered our third line of defense. This type of immunity is specific, as I mentioned before, rather than non-specific like innate immunity is, and there are specific cells that have adapted to kill specific microbes within the body. Adaptive immunity is orchestrated by two different classes of white blood cells called lymphocytes. And the two major classes of lymphocytes are B lymphocytes, also known as B cells, and T lymphocytes, also known as T cells. As we will discuss shortly, B cell mechanism is oftentimes classified as antibody-mediated immunity. And this type of immunity is sometimes called humoral immunity. Humoral, humoral immunity. T cell mechanisms are often classified as cell-mediated immunity, and this is often referred to as cellular immunity. So we have humoral immunity and we have cellular immunity. Now, referring or relating to the image on the screen here, I want you to recall from our last lecture that lymphocytes originally form in bone marrow from primitive cells called hematopoietic stem cells. T cells actually mature in the thymus, but B cells mature in the red bone marrow, among other areas that are not discussed on this slide. And they mature in these areas before they're actually released into the circulation and end up traveling to lymph nodes as well as the spleen, which is not indicated on this diagram. Adaptive immunity requires the activation of these B cells and these T cell populations, and at this point it will begin their immune attack of specific antigens. Such activation is going to require two activating signals, so signal 1 and signal 2. It's going to require these two activating signals are a specific antigen, which you can see here, as well as a chemical, a molecule that's released from infected or injured tissue cells. You can see here that each lymphocyte has receptors for antigens and receptors for these signal chemicals. And what this process does, is it activates the immune cells, whether it's a B cell or a T cell. And we'll speak even more about this a little bit later. Now, as I've already told you, B cell immunity is also referred to as humoral immunity, and that B cells develop in red bone marrow. And at this point, they are referred to as a naive B cell. After completing, sometimes you'll hear them called almost like a virgin B cell, but for this course we're referring to them as naive B cells. And this is what they're called after they complete their first developmental stage. We call them naive or inactive B cells. These naive B cells synthesize antibody molecules, okay? So naive B cells are going to actually produce antibody molecules, but they don't secrete them. What they do instead is they insert these antibodies on their cell surface. Okay, remember we're talking just about B cells. They insert these antibodies on their cell surface, so on the plasma membrane. Once these B cells are released from bone marrow, these naive B cells, they travel to lymph nodes, they travel to the spleen, they travel to the liver. But activation of a naive B cell must be initiated by its encounter with a specific antigen, as I spoke to you about on the last slide. And this antigen has to fit perfectly with the antibodies on the cell surface. When an antigen binds to these antibodies on the B cell surface, activation of the B cell can occur. And of course, we also know, going back to the last slide, that it also needs a second signal, which is a molecule from the infected tissue, or from the microbe itself. Okay, so just once again, when the antigen binds to the antibodies that are found on the B cell surface, activation of the B cell occurs, and this triggers a very rapid series of mitotic divisions or cell divisions. And essentially, this rapid mitotic division creates a clone. And the clone will differentiate into such cells as a plasma cell, also called an effector B cell, and a memory cell. And the plasma cells are going to, we'll talk about each of them independently for a moment. The plasma cell is going to synthesize and secrete huge amount of antibodies. Huge amounts of antibodies. And again, they're also called effector B cells. And memory B cells, they don't secrete antibodies, but they have a very important role. So memory cells will hang around, and if they're exposed again at a later time to the antigen, the same one that triggered its initial formation, then it's going to rapidly divide, go through rapid mitotic divisions, and produce more of these plasma cells and more memory cells. Now let's talk for a few minutes about an antibody structure. Antibodies are proteins of the family called immunoglobulins, and a lot of times you'll see these antibodies or immunoglobulins as written as Ig, capital I, small g. Now, like all proteins, antibodies are very large molecules and are composed of long chains of amino acids, which we call polypeptides. And each immunoglobulin molecule consists of 4 polypeptide chains. 2 are heavy and 2 are light. And the chains are folded in such a way to form globular regions that are joined together in a way that the immunoglobulin is actually forms into like a Y shape. And you can see in this diagram, you can see that there are 2 twisted red strands, which are called the light chains. And you can see that there are 2 twisted blue chains, which are the heavy strands. And the heavy strands are heavier, as you would imagine, but they're also longer than the light strands. Immunoglobulins contain what are called variable regions, which are regions where the sequence of amino acid varies in different antibody molecules. So I'm referring to the sequence here. Okay, so the sequence varies. That's why they're called variable regions. So it's just represented by these colored bars that you can see in Figure B. Okay, variable regions through here. These regions can take different shapes, which can bind to different types of antigens. So you can think of different naive B cells, which contain different surface antibodies, can bind to different microbial agents. Because as I told you earlier, naive B cells contain a lot of different antibodies that sits on their surface. We'll spend just a moment talking about the clonal selection theory, which deals with antigen destruction, so destruction of that foreign substance. And this clonal selection theory holds 2 basic tenets. First, it holds that the body contains an enormous number of diverse clones of cells, so many clones of different cells in our body that are each committed to synthesize a specific antibody to attack or attach to a specific antigen. Secondly, when an antigen enters the body, the antigen itself knows to select the clone whose cells are committed to synthesizing its specific antibody. And it stimulates these cells to proliferate and produce even more antibodies. So when the antigen comes in the body, it's searching for a specific cell that contains its corresponding antibodies. And this will then further stimulate the cell to proliferate and produce even more antibodies than were already there. And I had mentioned this when we first introduced B cells and spoke about plasma effector cells and memory cells a few slides back. The graph on this page depicts response times, or antibody response times. And as you can see, the initial encounter with a specific antigen, which is the primary stimulus, you can see on the far left of this graph, is what's going to produce the primary response, okay, the first response. And as you can see in this diagram, after so much time that our body has been exposed to this antigen, we're going to start producing immunoglobulin M and immunoglobulin G. And this happens usually over the course of the next few days. Once the antibody is dealt with, you can see that there is then a drop in both, returning to baseline. Now remember earlier I told you that memory cells hang around, memory B cells hang around, and they're just waiting around for what we call a subsequent exposure. So a later encounter, so in this case a secondary stimulus, a later encounter with the same antigen will trigger these waiting memory cells to act much faster than the response was before, and they can at that point even produce more memory cells and more plasma effector B cells and release even more antibody than they did before. So this later encounter, as I said, is called the secondary response, okay, from a secondary stimulus. And you can see that both immunoglobulins respond much faster. There's much more antibody there than they did to the first exposure with the antigen, because now it recognizes it and it can work harder and faster. IgM and IgG are just specific antibodies that are released in certain exposures to antigens. And we'll speak about the different forms of immunity now on the next two slides, comparing natural versus artificial and active versus passive immunity. Okay, let's move on to the next slide where we'll talk about these different types of immunity briefly. So first, I'm comparing different types of natural immunity. We're comparing active first with passive immunity. I'm going to just speak of this lower part of the slide here. A natural active infection means that your body is naturally able to produce antibodies when exposed to an infection. So the active component is your body actually actively producing the antibodies. And it's natural because it's our body's natural method of doing it. It knows how to do it naturally. Natural passive acquisition is a little bit different. Natural meaning your body is still able to naturally do this, but passive refers to the case of a mother breastfeeding or a mother that's pregnant with a baby passes on her antibodies into the baby's system naturally. But it's a passive process because the baby is not actually producing the antibodies. Looking on to artificial immunity, we can either have artificial active immunity or artificial passive immunity. So in the case of artificial active immunity, a good example is immunization or vaccines. So what we're doing with vaccines is we're injecting the body with pathogens that are no longer virulent, meaning that they're not able to cause the disease because it's been treated in such a fashion that it can't. And so when you inject your body with this vaccination, then your body is able to produce antibodies against the antigen. So it's done actively, yes, because we're producing our own antibodies, but it is artificial because it's being injected into the body. And then lastly, artificial passive immunity. This is a situation here where you're actually injecting the antibody itself into a person. So it's done passively because, again, you're not producing the antibodies yourself, and it's done artificially because it's being put into the body, and it's been produced and manufactured outside of the body. And an example of this would be injection of gamma globulin. And gamma globulin can help people that have been, let's say somebody reached into a bag and was pricked with a needle, and there was the uncertainty as to whether or not that needle carried an infectious disease. Then that person can be injected with an antibody to hopefully help them fight off any potential infection that they may have acquired. Okay, so in terms of antibody function, the function of an antibody molecule is to produce antibody-mediated immunity, which we know is a type of immunity also called humoral immunity. And for interest's sake, the reason it's called humoral immunity is because it occurs within the plasma, which is one of the humors or fluids of the body. Now, essentially, antibodies act on antigens by inactivating and bending them together to facilitate phagocytosis, and by initiating inflammation and activating the complement cascade. Just as a reminder, the complement proteins, that group of enzymes that produce a cascade of reactions resulting in a variety of immune responses. So let's take what I just said and apply it to the diagram here and maybe even elaborate a little bit more. Okay, so as you can see in this diagram, we have an antibody, which right here, an antibody, and we also have an antigen. And this antibody is going to recognize whether or not this antigen is self or non-self, or so harmful or harmless. And when it's recognized as foreign, it can then produce one or more of its effect. So what will this effect be? Well, it may just inactivate the antigen, transforming it into something not harmful. It may also cause agglutination, which you can see here. And agglutination is just the clumping of these antigens, the antibodies clumping the antigens together. And this actually puts it in a nice, easily accessible position so that macrophages and other phagocytic cells can perform phagocytosis by eating and digesting the foreign product, right? So you can see here the facilitation of phagocytosis. The binding of the antibody can still produce yet another effect, which is activating that complement cascade that I spoke to you about before, that contributes to the destruction of foreign cells. And this leads to inflammation, chemotaxis, and lysis, which you can refer back to earlier slides if you need to review. And we have learned that mast cells are also involved in the inflammatory process, so antibodies can actually become, antibodies that are attached to them can have antigens. Antibodies can attach to these mast cells, and then the antigens can attach to the antibodies, which then initiates a inflammatory reaction with chemicals in the body. And we spoke a fair amount specifically about B cells, and now let's elaborate on T cells. T cells initially develop in red bone marrow, and then they mature, so they migrate to the thymus, where they mature in the thymus, and then they're going to migrate to lymph nodes as well as to the spleen. When an antigen encounters a naive T cell, so a naive T cell, similar in name as the naive B cell. So this is a T cell that has receptors on its surface. If you remember back, if I go back for a moment to the B cells, I had said that B cells have all of those antibodies covering their surface. That's the B cell. But with a naive T cell, it's a little bit different because it actually has antigen receptors on their surface. So similar in terms of function, but different. The antigen binds to the T cell receptor when it's exposed to it, and this causes activations, or what we call sensitization of the T cell. And this leads to very rapid mitotic divisions, just like we spoke about with B cells, which transforms it into, or divides into, different T cells that are now specific sensitized T cells. And the types are effector T cells and memory T cells. Effector T cells are going to secrete cytokines, a variety of different cytokines, and memory T cells are going to do just like what they did with memory B cells did, which is remaining in the body until they're needed to produce an additional effect, which is going to be, okay, they now encounter another antigen, they recognize it, then they stimulate the production of more effector T cells and more memory T cells when this is needed. As I already mentioned to you, T cells display antigen receptors on their surface membranes, which is different than the antibodies that are present on the B cell membranes. So it's different, but there is a lot of similarities. T cells can only react to a foreign antigen that has already been phagocytosed and then presented on the surface of an antigen-presenting cell, meaning that once the foreign antigen has been already phagocytosed, that cell is going to present the proteins on its surface as an antigen-presenting cell, and now the T cells can react with this cell. And we're going to form what's called a junction between the T cell and the antigen-presenting cell, which forms it's a temporary junction called an immunological synapse. Okay, so of the effector T cells that I just spoke to you about on this slide here, of the effector T cells, which secrete a variety of different cytokines, are cytotoxic T cells. And these cause what's called contact killing of a target cell. So sometimes they're also referred to as killer cells. And you can see in this diagram on the right here that cytotoxic T cells, which are also referred to as killer cells, can actually attack cancer cells. Okay, so we have a cancer cell here, and T cells you should think of T cells as being a very significant part of our defense against cancer and other abnormal and foreign cells as well. But after forming an immunological synapse with the tumor cell, the cytotoxic cells are going to release perforin. You can see these perforin molecules. And essentially, skipping a few steps in the process, what these perforin molecules do is they perforate the cancer cells, but then they end up triggering apoptosis of these cancer cells, which is that programmed cell death. So it basically programs them to die. And on the last slide here, this figure summarizes the stages of adaptive immune response, and we'll go through it just briefly because a lot of it's review. So first of all, the B cells and the T cells here are going to recognize a specific antigen. Okay, the recognition phase. Next, the B and T cells are activated in the activation stage. And when they're activated, this is when we're going to see a lot of expansion of their population. So we call that cloning. So it'll differentiate into those effector cells and the memory cells that we spoke about earlier. Then the effector cells, which are working to take effect, are going to get to work by attacking the source of the antigen, so attacking the antigen. And that's during the effector phase, with the purpose being to eliminate the antigen. This is done through both humoral immunity and cell-mediated immunity, of course. Now, as antigen levels begin to decline, effector cells are going to start to die off through the process of apoptosis. So our own cells are going to die through programmed cell death because they're not needed anymore. But what we do know is that we have those memory cells that remain, and those memory cells are going to be surviving and waiting around, ready for possible invasion of that antigen again. And if that antigen does invade again, then of course we know the next step is going to be these memory cells producing more of the effector cells and more of the memory cells producing much more antibody than was there in the beginning to increase our odds of fighting off whatever infection or pathogen has invaded our body. That's the end of today's lecture. We'll see you in class next week.